Okay, amazing AP Bio 2 kiddos, are you ready for this video, which is the continuation of Unit 4, Cell Energy? This video, the topic shall be Oxidative Respiration. However, I thought I would start on this last slide that we were talking about in class. I kind of wanted to just recap glycolysis before I get into oxidative respiration. So I know that this is me repeating because I've already talked about it, but I just thought that we would be able to put this into perspective before we get into the heart of oxidative respiration. Okay, so just really quickly, summary of glycolysis. Let's see what you guys remember. Where does it occur? Hopefully you said cytosol. And this is in all organisms. It doesn't matter if it's prokaryotic or eukaryotic. This process of glycolysis, which is sugar breaking or sugar splitting, will happen in the cytosol under anaerobic or aerobic conditions. Technically, this process is anaerobic, which means it does not require oxygen. However, if oxygen is there, it will also continue to occur. Okay, but it is known as an anaerobic process, this first step in cellular respiration. Okay, just to recap also, we talked about in class how there was an energy investment phase to glycolysis, and then there's an energy payoff phase. The investment phase was the utilization of 2 ATP in the process of splitting glucose, but the payoff is we're going to form 4 ATP, so if we use 2, okay, that's -2, take off those, we gain 4, 4 minus those 2 will give us a net gain of 2 ATP overall. And remember, the gain of those 2 ATP is through substrate level phosphorylation. Okay, substrate level phosphorylation, which means there's the utilization of an enzyme, a substrate molecule, and that phosphate will be pulled off of the substrate to form our 4 ATP. Okay, what else do we gain? Remember, at the end of the process, we will have 2 pyruvates or pyruvic acids. Okay, and once we do that, to be able to split that glucose, we're going to have some oxidation. And if we oxidation, we're going to oxidate it and gain electrons. So the reduction, because remember these are redox reactions, the oxidation is of the glucose, the reduction is of these NAD pluses, and once they're reduced, they become NADH, and these are our electron carrier molecules, remember, that are going to play an important role in going to the electron transport chain. The goal of them is to go to the ETC, where they can be utilized through oxidative phosphorylation and chemiosmosis to gain the most ATP. Okay, so in glycolysis, we start with glucose molecules. We're going to use a little bit of ATP, 2 of them. We're going to split the glucoses into pyruvates or pyruvic acids. We will gain a net of 2 ATP, and we will get these electron carriers known as NADHs that will go to the electron transport chain. Okay, so overall, if you look at the bottom, this is the net, okay, what we gain out of this. 2 NADHs, 2 ATP through substrate level phosphorylation, and we gain 2 pyruvates that are going to now go into the mitochondria and go to the next step. So if you look at the overview, I really love this picture. This diagram is amazing because it kind of puts it all into perspective. You can see that glycolysis is step 1, and this is out in the cytosol, and we will utilize ATP, but we will gain 2 ATP through substrate level phosphorylation. We will get pyruvates or pyruvic acids. There will be 2 of them that now will head into the mitochondria through active transport. They will be pumped into the mitochondria, and at that point we will continue to go through oxidative respiration, which entails Krebs Cycle and electron transport chain. Okay, so we have to continue the oxidation of pyruvate, which is why we must go through Krebs Cycle, and then at the electron transport chain, this is like the parte of all parties, we are going to generate the most ATP through the electron transport chain, because this will utilize oxidative phosphorylation with chemiosmosis, and that will give us the most bang for our buck. Okay, so if you look at glycolysis, pyruvic acid or pyruvate are going to go into the mitochondria. Look at our NADHs that will go up and over and go to the ETC. But right now what we're going to focus on is oxidative respiration, which will happen here in the mitochondria. Okay, before we get into that though, I do want to give you 2 names. If I point to this space inside of the mitochondria, which is a jelly-like consistency, the name for that is matrix. Okay, it's kind of like the stroma inside of the chloroplast. So it's the gel-like fluid inside the mitochondria is known as the matrix. Okay, the actual membranes right here and here that fold back and forth on the inside of the mitochondria, those are called cristae. It's really important that I go over those two terms with you because in a second, when I get into the Krebs cycle and the grooming step, those will occur in the matrix, whereas the electron transport chain happens on the cristae. So the location is very important, kind of like what we did with the chloroplast when we talked about the light reactions happening on the thylakoid, whereas Calvin cycle happened in the stroma. This is, you're going to notice, the Krebs cycle, which is kind of like the Calvin cycle, only it's opposite. It's going to be happening in the liquid portion, which is the matrix, and the electron transport chain happens on the cristae. So it's kind of like the ETC and the chloroplast happening on the thylakoid membrane, only in the mitochondria it's happening on a cristae, because in order to generate the most ATP through that chemiosmotic process, you must have a membrane, which is the cristae in this case. Okay, so now let's get into the heart of this, oxidative respiration. Okay, the overview, just to let you know, we're going to take the two pyruvic acids that are from glycolysis because they still contain a tremendous amount of chemical energy. Now, if you say, really, how much chemical energy is there? When we went through glycolysis, we released less than a third of the chemical energy that is contained in that original glucose. Less than a third is tapped into and oxidized during glycolysis. So we have a ton more chemical energy that is still in the bonds of these 3-carbon, remember these are 3-carbon, pyruvates or pyruvic acids that we must continue to oxidize to release the electrons so we can tap into that for our bang for our buck to get the most ATP. So here's the big if oxygen is available in the mitochondria, okay, now when would it not be available? If you were exercising vigorously, and we're going to talk about that later in this unit. But under normal conditions, if plenty of oxygen is available inside the mitochondria, those pyruvates or pyruvic acids will enter into the mitochondria, will go into the matrix area, and will be further oxidized, okay? But before we get to Krebs, which is where we're going to do most of our oxidation, put a big star right here, there is a grooming step that must happen prior to entering the citric acid cycle or the Krebs cycle. So this grooming is really important because we're going to take a 3-carbon molecule, which you know is pyruvic acid, pyruvate, and we're going to break it down into a 2-carbon, what we call Acetyl Coa. So we have to do this little grooming step. This isn't the complete oxidation, it's like a pre-oxidation, but we're going to ultimately take a 3-carbon pyruvate and break it into a 2-carbon acetyl CoA. Well, if I break a 3-carbon into a 2-carbon, I'm going to lose a carbon. How do you think I'm going to lose it? Look what it says right here. We're going to give it off as CO2. That's a gas. How do we give off a gas? Well, you guys should remember it's going to diffuse out of the mitochondria, jump into the cell, into the cytosol, diffuse out of the cell, probably jump on a red blood cell and make its way to the lung where you can exhale it and get rid of it as a waste product. So this grooming step, also known as the intermediate step, is where we're going to take the 3-carbon pyruvate, break it into a 2-carbon acetyl CoA, give off the carbon dioxide as waste product, okay, and then we're also going to have a little reduction. What are we going to reduce? You guys know we reduce these electron carriers. If I break off a carbon, if I break it off and lose a carbon, I'm releasing electrons. Those electrons are picked up, in this case, by NAD+. That's our electron carrier. So that will reduce it to NADH. Remember, we also get some hydrogen ions because it doesn't pick up all of the hydrogen ions. This will head to the ETC, like it normally does. Okay, I now have this new acetyl-CoA molecule that will go into Krebs cycle. Now, if you want to know, well, how does this become acetyl-CoA? The 2-carbon molecule is known as acetate that will bind to a coenzyme. We have to use a coenzyme. Remember, a coenzyme is a catalyst, and it speeds up the rate of a reaction. So this coenzyme A is going to help out by binding to the acetate to facilitate this process, so we're going to call this acetyl-CoA. Okay, now to show you the diagram of what this will look like, this is a great picture. Okay, so let me write on this a little bit. You can see this is the cytosol. Here's the mitochondrial membrane, so here's the mitochondrion. So right here, this is known as the matrix inside the mitochondria. So here's our pyruvate, or pyruvic acid. There is a group right here, you probably remember it from our biochemistry unit. This is the carboxyl group. Okay, that carboxyl group, sometimes you guys know it's written as C double bonded to O, and then the C is bonded to an OH. Well, the H is already lost in this case. Okay, that H has already been taken off of this. So it's in ion form, but this is known as a carboxyl group. So as the pyruvate is transported into the matrix, and remember this is active transport, once that is pumped into the matrix of the mitochondria, okay, number one is this carboxyl group is broken off to form our carbon dioxide. That's our waste. Okay, number two, we have the oxidation, and as the oxidation occurs, we have electrons that are picked up by NAD+, remember 2 electrons. It also picks up hydrogen, and then some of the hydrogen is released out here into the matrix area. So we're going to have hydrogen ions built up into the matrix. Okay, so this now NADH becomes an electron carrier. Where do you think he's headed? Hopefully you said headed to the ETC, because that's where all electron carriers go. Okay, now we have coenzyme A that comes in the third part, which is a sulfur-containing compound. That's where this S comes from. Coenzyme A is a sulfur-containing enzyme. It bonds to our 2-carbon molecule. If you want to know, well, why is this 2-carbon? Remember, we lost a carbon over here as carbon dioxide, so that comes off. We now have a 2-carbon molecule. You can see it in pink. That 2-carbon molecule bonds to this coenzyme A, sulfur-containing enzyme, to become acetyl Coa. Okay, this is known as the grooming step. What it accomplishes is it accomplishes the oxidation of this, which means the extraction of electrons. It also accomplishes, we lost the carbon dioxide as waste product, and now we have something, this acetyl Coa, that is ready to go to the Krebs cycle. Okay? All right. Okay, now we're ready to move into the Krebs cycle. Okay, so the next slide, there's a few names I want to make sure that you understand can be used, although I tend to use Krebs cycle, which you can see in the middle of this diagram. That's my preference for the word, but some books and some teachers will constantly refer to it as the citric acid cycle. I'll show you why in just a second, because that's the name of one of the compounds that's formed in the cycle. Some people call it the tricarboxylic acid cycle, TCA cycle. I tend to use the old school Krebs cycle, which the name comes as honoring Hans Krebs, which is the German-British scientist who was largely responsible for working out the pathway in the 1930s. So I tend to give him, Hans Krebs, a little credit and use that, although a lot of teachers use citric acid or TCA cycle. Okay, so we are partaking in oxidative respiration. Remember, we are in the mitochondria, and specifically, we are in the matrix. We are in the liquidy portion to undergo this. I kind of want to show you in this diagram, however, from here to here, if you want to circle this on your notes, and this is the grooming. So it's showing what I just went over in the last slide. This is the grooming step. They included it in the diagram. So you can see our pyruvate, which is from glycolysis, will be broken down, the pyruvate, which are from glycolysis, which are 3 carbon molecules, are converted into 2 carbon molecules in this process of grooming. Okay, and we have 2 of these, don't forget. So like there are 2 pyruvic acids, which is what this is showing you. So here they are. This is a 3 carbon. There's 2 of these that will go through the grooming step, which is the removal of one of the carbons as carbon dioxide. The use of coenzyme A, the sulfur-containing compound, comes in, okay, is going to bond with that to become acetyl Coa, and in the process, a little oxidation occurs. And when we oxidize, the oxidation of the molecule is going to help reduce this electron carrier. So the NAD+ becomes reduced as NADH in the oxidation process. Okay, we also gain some of these hydrogen ions that are now inside the matrix. Okay, now we're ready to proceed to the Krebs cycle, which you see down here. So we're going to start Krebs with acetyl Coa, but remember there are 2 of these because our original glucose was split into 2 3-carbon molecules. So now the acetyl-CoA, if you look at this, comes into the cycle. The CoA, which is the enzyme, breaks off and is recycled and can be used over again. Okay, this is the recycled part because it's an enzyme, and enzymes are never used up in a reaction. Okay, the remaining molecule, this 2-carbon molecule, comes in. There's already something in the cycle over here. You guys know cycles constantly turn. There's something over here in the cycle. I call him Big O. His name is actually oxaloacetate. You'll see it in the next picture. Okay, oxaloacetate. Oxaloacetate is already in the cycle, will join to this 2-carbon compound and form a 4-carbon molecule over here known as citric acid. That's where the name citric acid cycle comes from, because the compound that's formed here in this Krebs cycle is known as citric acid. That will then go through a series of changes along the way. As we go through the series of changes, you'll notice the 2 carbon dioxides break off. That is ultimately these 2 carbons that are left from the original glucose. So we will lose the carbon dioxides, but in the oxidation process, we're also going to reduce some more NAD pluses to become NADHs. How many? Well, for one of these acetyl-CoAs coming in, for one of them, we get 3 NADHs. Over here, we're going to have another electron carrier known as FADH, who is only used in Krebs. Okay, he is also, with FAD, reduced to FADH too. That will come out of this. For my 1 acetyl-CoA that comes into the cycle and goes around, I will gain 3 NADHs, 1 FADH2, but I will also gain 1 ATP. That's for 1 spin of Krebs cycle. But remember, this thing is going to spin twice because we had 2 pyruvic acids from the beginning that were groomed, so we had 2 acetyl-CoAs. So if you do all the numbers, from my original sugar, my original glucose, Krebs will spin twice. So my total gain will be 6 NADHs, 2 ATP, and by the way, this is through substrate level phosphorylation, just like it was in glycolysis, and we will gain 2 FADH2s. So this picture is showing you 1 spin of the cycle for 1 acetyl-CoA, but you have to remember to tell yourself it's spinning twice because we have 2 acetyl-CoAs. Okay? So after this cycle is completed, what you ultimately have, which is amazing, is we will have the complete oxidation of all of our sugar molecules, because we will lose them as these carbon dioxides. We'll oxidize them and gain the electrons to go to the electron carriers that will then go to the ETC. That's where all electron carriers go. So let's look at the bullet points over here on the side and make sure you understand this. This occurs in the matrix of the mitochondria. The production, put a star by this, what we're producing, we will get NADHs, FADH2s. How will we get that? From the oxidation of our acetyl-CoAs. We will produce ATP, and we will release carbon dioxide as our waste product. Okay, CoA, coenzyme A, is not actually part of the reaction. It is recycled, so you can see that arrow coming off here. So remember, it is an enzyme, it is recycled. Now, if you put numbers with these, from a full glucose, okay, in the beginning that went through glycolysis, okay, we would have 2 pyruvates, which means we have 2 acetyl-CoAs. So if we put numbers with this, it should be 6 NADHs, 2 FADH2s, that's just in Krebs. Okay, don't forget we also have this NADH right here that's gained. Okay, and then we get ATP will be 2 from our spinning twice, and we'll get carbon dioxide. We get 4 from just Krebs. But don't forget, we had 2 up here from the pyruvate that was forming the acetyl-CoA, because that was 2. So if you add 2 to that, that should be 8 NADHs, 2 FADH2, and this would be 6 CO2. If you take the grooming step with the Krebs, that's what you gain. Does that make sense? All right. Okay, so the last slide, and then we will complete Krebs cycle, or citric acid cycle. I really like this diagram because it shows you the intricate details. Oops, I forgot to go on full screen. Hang on. There we go. I really like this diagram. However, I'm not going to hold you responsible for memorizing all of these specific names of the compounds, so don't freak out on me with that. I kind of just want you to understand the philosophy and how the cycle is running. Okay, so this is a closer look at citric acid cycle. So in the beginning, what you see here, we have our Acetyl Coa, but you have to remember, keep telling yourself, we really have two of these because we had the two pyruvic acids that went through that grooming step. So this grooming has already occurred in this picture. Okay, so in this picture, grooming has already happened. And so what you see are the acetyl-CoAs, which is a 2-carbon molecule, comes into the citric acid cycle. Remember, there's already something in the cycle, and here he is right here. I call him Big O. Don't ask me why, I just have always used that. His name is Oxaloacetate. He is a 4-carbon compound that is already in the cycle. It's kind of like when we did the Calvin cycle, and remember we had the RUBP or RUBIP that was in the cycle that constantly was there. It's the same philosophy. We have this oxaloacetate, a 4-carbon compound that is already in the cycle. So it comes together. Okay, joins with this 2-carbon acetyl-CoA, so 4 and 2 makes a 6-carbon compound known as citrate or citric acid. Now here's where you'll notice the coenzyme A comes out of this. Once the acetyl-CoA bonds to the 4-carbon compound, the coenzyme A is recycled again. But now we have this 6-carbon compound, but remember we have to take those 2 that are from acetyl-CoA, we need to get rid of those. And that's where we're going to get rid of them as carbon dioxide and carbon dioxide. And so the goal is to get rid of those and then take the 4-carbon compound and be able to produce it over here again and recycle it as oxaloacetate. So in the process of doing this, you can notice there's a lot of specific names. Like I said, you don't have to memorize all of these. I would like you to know oxaloacetate, and I would like you to know citrate or citric acid. But as far as understanding all of these other names, I just want you to know that these go through a series of oxidation-reduction reactions to extract the electrons. That's what we're trying to do. Extract electrons. Why? Because we're going to add those electrons to our electron carriers, NAD+, to reduce it as NADH, which can then go to the ETC. Why do we need these to go to the electron transport chain? Because remember, at the ETC is where we will go through oxidative phosphorylation with chemiosmosis, and we will generate the most ATP in that fashion. So to do that, we must use these electron carriers to get the electrons there. So the job of the Krebs cycle is to go through these oxidation-reduction reactions to be able to oxidize the compound, but in doing that, we have isomers that come out of this, like isocitrate is an isomer of citrate and then goes through a series of other changes. But in doing that, we're going to get NADH, okay, NADH, you see another one over here, NADH. We will also get FADH2. We will lose carbon dioxide as a waste product, and then we will gain ATP. So from this one spin, okay, one spin of this cycle, just look at this diagram, it's just showing you one spin. We get 1, 2, 3 NADH. We get 1, 2 CO2. We get 1 FADH2. And we get 1 ATP. That's for one spin. But we have to take 2 spins because we have 2 acetyl-CoAs. So if you double those numbers, you'll see what your results are. And at the end of this cycle, we are ready to go to the electron transport chain, or we can go through oxidative phosphorylation. And I'll do that in another video. Okay? Make sense? Great job.